Dosage compensation. So what is dosage compensation? Actually, female contain two X chromosomes while male carries single X chromosome along with a Y chromosome. So obviously there is a difference of genetic dosage regarding the X-linked genes. Female should produce more product of X-linked genes because female carry two X chromosome while male carry a single X chromosome, so male produce a single product for all X-linked genes. So how the extra dosage problem is compensated in both male and the female. So one X chromosome of female become inactivated, and in that way extra dosage problem is compensated. So, dosage compensation is to balance the genetic product of X chromosome in both sexes, in both male and the female. Barr and Bertram are the two scientists who worked on the female cats, and they observed in the nerve cells of female a special dark stained body. So that specific body is absent in the same cells of male cats. While Moore and Barr further carry on their experiments on human female, and in the human female, they work on the fibroblast cells and they observe a same dark stained body in the human female cells. But that darkly stained body is absent in the fibroblast cells of human male. So they named that darkly stained body as Barr body. Barr body diameter is approximately 1 micrometer, and that Barr body is the darkly stained body present only in the female mammals. And it was observed in the female cats, in the human female, and some other mammalian females. So Ono in 1961 presented his hypothesis that as female carry two X chromosomes, so one X chromosome of female become inactivated, and that inactivated X chromosome forms the Barr body. So Barr body is actually a condensed form of X chromosome. Out of the two X chromosomes of female, one X become inactivated, one X become condensed, and that inactivated X chromosome converted into the Barr body. So, in that way, when one X become inactivated in the female, it means at a time only single X chromosome of female is forming its product. So this was shown by culturing cells with different karyotypes. For example, as male carry single X chromosome along with Y. Similarly, in case of Turner syndrome, when single X chromosome is present along with autosomes, and you can say single X chromosome is present and its partner is missing, means single sex chromosome is present in that female. So both of these cases are with one X chromosome. Similarly, the case in which female carry two X chromosome and that is a normal female, and the case of Klinefelter syndrome when two X are present along with a Y. So double XY, that is a condition of Klinefelter syndrome, and Klinefelter syndrome is present in male. So both of these cases carry two X chromosomes. Similarly, triple X, tetra X, these carry 3 or more than 3 X chromosomes. So how can we calculate the number of Barr bodies in these cases? The formula for the calculation of number of Barr bodies is n minus 1, where n represents the total number of X chromosomes. So out of the total number of X chromosomes, we have to minus 1. So the result is the number of the Barr bodies. For example, in case of XY, a single X chromosome is there, so we have to apply the formula and n is equal to 1. So 1 minus 1 is equal to 0. So it means in case of XY, no bar body is present. Similarly, the next case is X0, and in that case also no bar body is present. In case of double X, two X chromosomes are there, so according to the formula, the number of bar body should be 1. So 1 bar body is present in double X. Similarly, we can calculate the bar bodies in other cases. So triple X individual should contain 2 bar bodies. It means only one X chromosome is active in any given cell, the other is inactive. According to the mechanism of the inactivation, according to the bar body formation, at a time one X chromosome is active, other is inactive. So in some cells, the paternal allele is expressed, and in other cells, the maternal allele is expressed. What is meant by that? As you know, female carry 2 X chromosomes. So out of these 2 X chromosomes, one X has maternal origin. It means it comes from mother, while other X has paternal origin. So one X has maternal origin, while other X has paternal origin. So maternal X comes from mother, paternal X comes from father. So if one X become inactivated, it means the other X will express itself. So that's why it was written here that in some cells the paternal allele is expressed, and in other cells the maternal allele is expressed. So in triple X and tetra X females and XXY males, only one X is activated in any given cell, the rest are inactivated. According to the formula of the bar body, as we have calculated in the previous slide. So in all these syndromic cases, at a time, there should be one X chromosome activated. So that figure is also representing the occurrence of bar bodies in various human karyotypes. And you can see here the normal male karyotype 46XY and the case of Klinefelter syndrome, Turner syndrome 45X. So according to the formula of the bar body, the number of bar body should be 0. Similarly, in the next case, 46XX, that is the normal female, and 47XXY, that is a Klinefelter syndromic male. So the number of bar body should be 1. Similarly, in triple X female and triple XY case, the number of bar body should be 2. It is indicating that 2 X chromosomes are inactivated. So it means in that case, 1 X is activated. 1 X will express itself only. Similarly, in case of tetra X and tetra XY, the number of bar bodies are 3. So question arises that if at a time only single X chromosome is activated, then why these cases are not normal? हमने पीछे बात की है कि अगर हमारे पास 2 X क्रोमोसोम्स होते हैं तो उसमें एक X क्रोमोसोम जो है वो एक्टिव होगा, दूसरा इनएक्टिव है. ठीक है, तो हमारे पास जितने भी सिंड्रोमिक केसेस होते हैं उसमें भी हमने यही बात की कि अगर हमारे पास जोन सा है यहाँ पे सिंड्रोम का केस है, क्लाइनफेल्टर सिंड्रोम है, जिसमें हम कह रहे हैं कि एक एक्स क्रोमोसोम इनएक्टिव होके बार बॉडी बना रहा है और दूसरा एक्स क्रोमोसोम जो है वो एक्टिवेटेड है. सिमिलरली नॉर्मल मेल और टर्नर सिंड्रोम इंडिविजुअल में कोई भी बार बॉडी जो सी है वो प्रेजेंट नहीं है. इसका मतलब क्या है कि इनमें क्या है, एक ही एक्स क्रोमोसोम है वो अपने आप को एक्सप्रेस करेगा. सो क्वेश्चन ये अराइज होता है कि अगर इन तमाम केसेस में एट अ टाइम जो सा है वो सिंगल एक्स क्रोमोसोम एक्टिवेटेड है तो ये एबनॉर्मल केसेस क्यों हैं? ठीक है, ये नॉर्मल क्यों नहीं है, ये सिंड्रोम क्यों कॉज करते हैं। फॉर एग्जाम्पल 45X का केस है जिसको हम टर्नर सिंड्रोम कहते हैं। इसमें सिंगल एक्स अपने आप को एक्सप्रेस कर रहा है। डबल एक्स वाई क्लाइनफेल्टर सिंड्रोम, सिंगल एक्स इज एक्सप्रेसिंग इटसेल्फ एट अ टाइम according to the formula of the bar body. Similarly, triple X, tetra X. So the answer is that X inactivation takes place in embryo at blastocyst stage, or you can say 32 cell stage. So X inactivation जो है वो embryo में होनी है और early developmental stages पे होनी है। और वो स्टेज कौन सी है? दैट इज अ ब्लास्टोसिस्ट स्टेज, or it is also known as 32 cell stage. It means 32 cells are already formed. So 32 cells में जितने भी X क्रोमोसोम्स हैं वो अपना एक्सप्रेशन दे चुके हैं, वो अपना प्रोडक्ट जो है वो बना चुके हैं। Next is that 15% of the X chromosome escapes inactivation. Means complete X chromosome will not be activated. Almost 15% of the X chromosome escapes inactivation and it will form its product. So the genes that are present on the 15% of the X chromosome express themselves and form their product. So Lyon hypothesis क्या है? 1961 में English geneticist Mary Lyon proposed his hypothesis to describe the mechanism of X inactivation. So his hypothesis and experiments consist on 5 tenets. The first is that condensed X chromosome is genetically inactive. जैसे हमने पीछे पढ़ा था कि X क्रोमोसोम बेसिकली कंडेंस्ड फॉर्म में होता है, इनएक्टिव हो जाता है और बार बॉडी बनाता है। सो दैट इज द सेम पॉइंट दैट कंडेंस्ड X क्रोमोसोम इज जेनेटिकली इनएक्टिव. Next is that X inactivation in humans occur early in development when embryo consists of about 32 cells. So 1 or 2 days following the fertilization. His next point is that at 32 cell stage or blastocyst stage, in each of the 32 cells, one of the X chromosome is randomly inactivated. ठीक है, ब्लास्टोसिस्ट स्टेज पे जाके ये जो 32 सेल्स हैं अब इनमें रैंडम X इनएक्टिवेशन होनी है। इसका मतलब क्या है कि आपके पास 2 X क्रोमोसोम्स हैं, वन इज द मैटरनल X, अदर इज द पैटर्नल X. So either the maternal or the paternal X become inactivated randomly in these 32 cells. Inactivation is mitotically stable. It means once inactivation takes place, then all progeny cells carry same inactivation. Like 32 cells में inactivation हो चुकी है, तो उसके बाद इन 32 सेल्स ने फर्दर माइटोसिस करनी है और इन्होंने फर्दर डिवाइड करना है। सो इनसे फर्दर जितने भी प्रोजेनी सेल्स बनेंगे उनमें सेम X क्रोमोसोम इनएक्टिवेटेड रहेगा। The next and last point is that net effect of this is to equalize phenotype in male and female for genes that are carried on the X chromosome. So what is the purpose of X inactivation? The purpose is the dosage compensation. We have to compensate the extra dosage problem for the X-linked genes in male and female. So the result is that the product for the X-linked genes equalize in both male and the female. So here is a mechanism. One X chromosome in female is inactivated early in the embryonic development. You can see here that here is a zygote that carry 2 X chromosome. One X is presented in blue color, other X in purple color. So you can say that one X comes from mother, other X comes from father. So random inactivation takes place and you can see here that in some cells, the X in blue color is inactivated and converted in bar body, and the other cells in which X in purple color become inactivated. So you can see here that some cells have uh, purple color X that express itself, and the other cells have X in blue color that will express itself. So further, these cells divide through the process of the mitosis, and the decision that was taken in the parent cells was mitotically stable, and same decision was passed in the progeny cells. So जिन cells में purple color का X जो है वो activated है, उससे बनने वाले जितने भी progeny cells होंगे, जितने भी daughter cells होंगे, उनमें same X जो है वो अपने आप को express करेगा. इसी तरह जिन सेल्स में एक्स ब्लू कलर का प्रेजेंट है, इसका मतलब क्या है कि वो ब्लू कलर का एक्स जो है वो अपने आप को एक्सप्रेस कर रहा है, और दूसरा एक्स जो है वो इनएक्टिव हो गया. तो सेम डिसीजन जो है वो इससे बनने वाले प्रोजेनी सेल्स में पास होगा. सो आप देख सकते हैं कि फर्दर जितने भी सेल्स बन रहे हैं, कुछ में ब्लू कलर का एक्स शो किया गया है और कुछ में पर्पल कलर का कौन सा है वो एक्स प्रेजेंट है. सो ये भी डायग्राम उसी को रिप्रेजेंट कर रही है. सो वी हैव 2x chromosomes. One X comes from mother, other X comes from father. He is a fertilized ovum. Mitosis हो रही है. Mitosis further जो है वो आपके पास blastocyst stage आ रही है. And in the blastocyst stage, both X chromosomes are activated. But later on at this stage, random inactivation of one X chromosome takes place. So in some cells, paternal X become inactivated. In other cells, maternal X become inactivated. So ये आपके पास अब एक सेल है और इसमें जो है वो maternal X inactive हो गया है. That is shown in green dot. जबकि ब्लू कलर का जो एक्स दिखाया गया है ये पैटर्नल एक्स है, मींस फादर ने ही एक्स जो सा है वो पास किया है. अब इस सेल से बनने वाले जितने भी प्रोजेनी सेल्स होंगे उनमें मैटरनल एक्स इनएक्टिवेटेड रहेगा एंड पैटर्नल एक्स विल एक्सप्रेस इटसेल्फ. सिमिलरली इन अदर सेल्स, इफ paternal X become inactivated, then maternal X will express itself and same decision was passed in all the progeny cells. Examples are present in female cats, and first example is the tortoise shell female. Tortoise shell is basically the coat color pattern that is present in female cat. Similarly, the second example is the calico female, and calico is also a coat color pattern that is present only in the female cat. So both of these phenotypes are present only in the female cats, and these female cats are present in heterozygous condition. So basically the coat color gene is present on the X chromosome of female, and as you know, female carry two X chromosomes, and if two X chromosomes of females are present in heterozygous condition and carry two different alleles of the gene that controls the coat color, then it can cause the tortoise shell female or the calico female. So no male cat carrying the tortoise shell and calico phenotypes. The reason is that males are hemizygous for the X chromosome. Male carries single X chromosome. So single X chromosome of male can carry only a single copy of the gene. And tortoise shell and calico phenotypes are present only in case of heterozygosity. So female carry two X chromosome, one X of female carry the orange allele, other X of female carry the black allele. So if X with orange allele is inactivated, so in these somatic cells, X with black allele will express itself. As you can see here that this is the picture of the calico cat and the tortoise shell cat. So you can see here the orange and black patches in that cat. So basically wherever the black color is present, ठीक है, जहां जहां ब्लैक कलर जो सा है वो शो हो रहा है, वो क्या रिप्रेजेंट करता है कि एक्स विद ऑरेंज एलील इज इनएक्टिवेटेड। जब एक्स विद ऑरेंज एलील इनएक्टिव हो गया तो एक्स विद ब्लैक एलील अपने आप को एक्सप्रेस करेगा। सिमिलरली जहां जहां पे ऑरेंज कलर आपको नजर आ रहा है वो क्या शो करता है कि एक्स विद ब्लैक एलील इज इनएक्टिवेटेड, सो इन दैट टाइम एक्स विद ऑरेंज एलील विल एक्सप्रेस इटसेल्फ। So the random distribution of orange and black patches in the calico cat illustrates the Lyon hypothesis. The white patches are present due to another gene. So the first diagram is the calico cat, while the next diagram is of tortoise shell cat. So in the calico cat and tortoise shell cat, what is the difference? That in the calico cat, white patches are also present, and these white patches are due to another gene that belongs to autosomes, while the orange and black color is due to the X-linked genes. So these white patches basically distinguish the calico cat from the tortoise shell cat. So tortoise shell cat only contain the orange and black patches, and this cat lacks the white patches. So ये random X inactivation को शो कर रहा है कि जहां जहां पे सोमैटिक सेल्स में X विद ऑरेंज एलील एक्टिव होगा वहां पे कोट कलर ऑरेंज है, और जिन सोमैटिक सेल्स में X विद ब्लैक एलील एक्टिवेटेड है वहां पे आपके पास जो कोट कलर कैट में है दैट इज अ ब्लैक। जबकि इसमें मेल कैट्स जो हैं वो टर्टोइस शेल या कैलिको नहीं हो सकती। क्यों? क्योंकि मेल कैट्स कौन से हैं, दे कैरी सिंगल एक्स क्रोमोसोम। सो सिंगल एक्स ऑफ मेल माइट कैरी द ऑरेंज एलील और इट माइट कैरी द ब्लैक एलील। सो अगर उस पे ऑरेंज एलील होगा तो ऑब्वियसली पूरे कैट के जो कोट कलर है वो ऑरेंज ऑरेंज होगा, और अगर उस पे जौन सा मेल के एक्स पे ब्लैक एलील प्रेजेंट है तो इसका मतलब क्या है कि मेल कैट का कलर ब्लैक होगा। Another example of X inactivation is the G6PD, that is glucose 6-phosphate dehydrogenase. That is a gene present on the X chromosome. This gene codes for an enzyme that breaks down sugar, as the name indicates. Basically, it is involved in the metabolism process. So females produce the same amount of G6PD enzyme as males. Although female carry 2 X chromosome, but 1 X chromosome of female become inactivated. That's why the product for the G6PD is equal in both male and female. So double XY, that is the case of Klinefelter, and triple X individual produce the same amount of G6PD as anyone else. What is a G6PD gene? Basically, G6PD gene, as the name indicates, that is a glucose 6-phosphate dehydrogenase gene, and there are various alleles for this gene. One is G6PD-A, and the other is G6PD-B. So these are the two forms of the gene. They produce different variants of the enzyme, and both of these variants break down the sugar. Female could be heterozygous for the A and the B alleles of the gene. So each cell only produces one of the two forms of the enzyme. So G6PD gene दो forms में है, एक आपके पास G6PD-A allele है and second is the G6PD-B allele. So दोनों allele enzyme की different forms बनाते हैं. So जो female heterozygous condition में है इसका मतलब उसके 2 एक्स क्रोमोसोम्स हैं, एक एक्स पे ए एलील है दूसरे पे बी एलील है। सो उस फीमेल के सोमैटिक सेल्स जो हैं वो एट अ टाइम जस्ट एक एंजाइम की फॉर्म बना रहे हैं। ऑल्दो उनके पास एलील्स दो हैं, ठीक है, वो दोनों फॉर्म्स बना सकते हैं एंजाइम्स की, लेकिन वो एट अ टाइम सिंगल एंजाइम बना रहे हैं। ये क्या इंडिकेट करता है? दैट इज इंडिकेटिंग द रैंडम एक्स इनएक्टिवेशन प्रोसेस। सो इट मींस only one of the two forms of the enzyme is formed by each cell, while the other X chromosome that carry the other allele that is responsible for the formation of other enzyme is inactivated at that time. Another example of the random X inactivation is the anhydrotic ectodermal dysplasia. That is a X-linked recessive disorder, and basically that is a disorder of sweat glands. Sweat glands are absent in that affected individual, and if male carry its allele on his X chromosome, so male is totally affected, while heterozygous female is carrier for the anhydrotic ectodermal dysplasia, but she is phenotypically normal. So here is a fertilized ovum that carry the maternal X, and that is represented in orange color, or while the paternal X is presented in green color. So early cell division takes place, and after that X chromosome inactivation takes place, and you can see here the random X inactivation. In some cells, paternal X become inactivated. In other cells, maternal X become inactivated. So further on, mitosis takes place. These cells further grow and form the progeny cells, and in the progeny cells, same decision was passed regarding the X inactivation. And you can see here that this heterozygous female has somatic cells, and these somatic cells are different situation regarding the X inactivation. So in this female, some somatic cells have skin lacking the normal sweat glands, while some sweat glands are normal. Means some somatic cells have normal skin, while some somatic cells have abnormal sweat glands, or you can say the laxa sweat glands. कि कुछ somatic cells में sweat glands absent हैं और कुछ somatic cells ऐसे हैं जिसमें sweat glands present हैं. अब बेसिकली ये फीमेल क्या है, ये फीमेल जोन सी है, ये हेट्रोजाइगस कंडीशन में है. इसके पास 2 एक्स क्रोमोसोम्स हैं. इसके एक एक्स क्रोमोसोम पे नॉर्मल कॉपी प्रेजेंट है, जबकि इसके दूसरा एक्स जोन सा है उस पे क्या है, उस पे म्यूटेंट कॉपी प्रेजेंट है या डिजीज कॉजिंग कॉपी प्रेजेंट है. अब क्योंकि एनहाइड्रोटिक एक्टोडर्मल डिस्प्लेजिया रिसेसिव डिसऑर्डर है, सो इट मींस हेट्रोजाइगस फीमेल इज फिनोटिपिकली नॉर्मल बिकॉज नॉर्मल कॉपी इज डोमिनेंट. लेकिन अब क्योंकि रैंडम एक्स इनएक्टिवेशन हुई है, तो जिन सोमैटिक सेल्स में एक्स विद नॉर्मल एलील इनएक्टिव हो गया, तो इसका मतलब क्या है कि उन सोमैटिक सेल्स में एक्स विद disease causing allele will express itself. So that's why वो somatic cells हैं उनमें sweat glands absent होंगे, जबकि वो somatic cells जिनमें X normal allele के साथ express कर रहा है, उन somatic cells में आपके पास sweat glands present होंगे और वहां पे आपके पास normal skin है. So that is another case of random X inactivation. X reactivation in females. So हमने पीछे पढ़ा है कि फीमेल में एक्स क्रोमोसोम इनएक्टिव होता है, बट एक्स क्रोमोसोम अगेन रीएक्टिवेट हो जाता है इन फीमेल्स. सो इन द फीमेल फीटस, future germ cells undergo lyonization along with somatic cells at the 32 cell stage. Following differentiation of female fetus, the inactivated X chromosomes are reactivated during female gametogenesis. जब फीमेल गैमीट फॉर्मेशन की स्टेज पे आएगी, तो उसमें इनएक्टिवेटेड एक्स क्रोमोसोम रीएक्टिवेट हो जाता है. ठीक है, फीमेल जब गैमेटोजेनेसिस की स्टेज पे आएगी, तो ऑल inactivated X chromosomes become reactivated. So when germ cells develop into oocytes and enter meiosis, their inactivated X chromosomes become reactivated so that every egg produced has an activated X chromosome prior to fertilization. So फीमेल ने जितने भी एग्स बनाने हैं, उन तमाम एग्स में एक्टिवेटेड एक्स क्रोमोसोम पास होता है. सिमिलरली एक्स रीएक्टिवेशन मेल्स में भी हो सकती है, और वो मेल्स कौन से हैं जिनमें क्लाइनफेल्टर सिंड्रोम है. क्लाइनफेल्टर सिंड्रोम दैट हैज अ karyotype double XY. So out of the two X chromosome, one X become inactivated, and that X also reactivate the during the gametogenesis. So male जब gametogenesis की स्टेज पे आता है, अगर क्लाइनफेल्टर सिंड्रोमिक मेल है और वो गैमीट फॉर्मेशन की स्टेज पे आता है, तो उसमें भी जो एक्स क्रोमोसोम इनएक्टिव हुआ था वो अगेन रीएक्टिव होगा. The presence of an extra X chromosome during early puberty causes death of male germ cells and testicular atrophy. But these male cells or these male are basically abnormal, and इनमें क्या होता है कि अगर एक्स्ट्रा एक्स क्रोमोसोम होता है तो वो अर्ली प्यूबर्टी पे ही क्या करता है, वो डेथ कॉज करता है, और मेल्स में टेस्टिकुलर एट्रोफी हो सकती है, और मेल्स में स्टेरिलिटी जोन सी है वो प्रेजेंट हो सकती है. सो दिस लीड्स टू लो लेवल्स ऑफ द टेस्टोस्टेरोन. एक्स्ट्रा एक्स क्रोमोसोम जो है वो टेस्टोस्टेरोन, दैट इज अ मेल रिप्रोडक्टिव हार्मोन, उसके लेवल को डिस्टर्ब करता है. The last point in the dosage compensation is mosaicism in humans. And what is mosaicism and how it occurs? The basic reason of mosaicism is the non-disjunction in which chromosomes are unable to segregate from each other. So what is its example? Its first example is the Turner syndrome, and second is Klinefelter's syndrome. So Turner syndrome, we have discussed previously, its karyotype is 45X0. Means total number of chromosomes are 45 in Turner syndromic individual, and that is present basically in female, and female carry only one X chromosome. While the Klinefelter syndrome, whose karyotype is double XY, total number of chromosomes are 47 in that individual, and what is the abnormality? That individual basically contain two X chromosomes along with a Y. So three sex chromosomes are present in Klinefelter syndrome, and Klinefelter syndrome basically affect the males. So these are the symptoms of the Turner syndrome or the Klinefelter syndrome. So what is mosaicism? Actually, we have discussing the mosaicism, and that is most common among insects. Sometimes one part of the body is male and the other part of the body is female. So that is producing the sex mosaics known as gynandromorphs or gynanders. अगर इंसेक्ट्स में बेसिकली ड्रोसोफिला में इसकी एग्जांपल जो है वो प्रेजेंट है कि ड्रोसोफिला में एक ऐसा केस देखा गया जिसमें ड्रोसोफिला की एक साइड, फॉर एग्जांपल राइट साइड अगर ड्रोसोफिला की है तो दैट इज मेल, एंड द लेफ्ट साइड ऑफ द ड्रोसोफिला इज फीमेल. सो अगर इंसेक्ट में एक पार्ट जो है वो मेल का है और दूसरा बॉडी पार्ट जो है वो फीमेल का है, सो दैट इज नोन एज सेक्स मोज़ेक्स. In human, when somatic cells show variation, like some somatic cells express paternal allele, other express maternal allele, इस condition को भी आप mosaicism कह सकते हैं कि different somatic cells are present in different situation, and that is basically due to the non-disjunction. Mosaicism जो है non-disjunction की वजह से आ सकती है, in which chromosomes are unable to segregate from each other. So what is the difference between intersex and sex mosaics? In sex mosaics, the boundary between male and female region is always sharp and distinct. ठीक है, सेक्स मोज़ैक्स में आपको क्लियरली डिफरेंस पता चलता है कि एक पार्ट जो है वो एक जो बॉडी रीजन है वो मेल है, एंड दिस अदर बॉडी रीजन इज फीमेल. Whereas in intersexes, all parts of the body may have a mixture of male and female characteristics, showing a condition intermediate between maleness and femaleness. So in intersex, you cannot distinguish between the male and the female characters. So the last is about the mechanism of inactivation, how X chromosome become inactivated and what is the mechanism behind that. The mechanism is the epigenetics, and epigenetics involve the two processes. First is the DNA methylation, second is histone deacetylation. So the whole process of inactivation is also known as genomic imprinting. Why it is known as imprinting? Because as we have discussed previously that once inactivation takes place, then same decision passed in all the progeny cells. So that is the imprinting. So basically the proximal end of the P arm of X chromosome has a region that is named as ZIC XIC, and that is approximately 1 megabyte in size. XIC is the abbreviation of X inactivation center, and that is basically participating in the inactivation process. That's why it is named as X inactivation center. It has 4 genes, and out of these 4, the most important is the ZIST gene. XIST is an abbreviation of X inactive specific transcript. ZIST, how it is participating in the inactivation. Basically, the ZIST gene is without open reading frame, ORF. ओपन रीडिंग फ्रेम जिस्ट में ORF एब्सेंट होता है. ORF क्या है? दैट इज अ ओपन reading frame that is necessary for the translation. Means once messenger RNA is formed, then messenger RNA should translate into the proteins or into the functional product. But due to the absence of ORF, RNA cannot translate. RNA cannot further form its product. So RNA of GIST cannot translate एंड कंटिन्यूसली अब जिस्ट क्या कर रहा है? जिस्ट जो सा है वो अपना आरएनए बना रहा है. ठीक है, ट्रांसक्रिप्शन हो रही है, जिस्ट का आरएनए बन रहा है, लेकिन वो आरएनए फर्दर ट्रांसलेट नहीं हो पा रहा. क्यों नहीं हो पा रहा? क्योंकि ओआरएफ एब्सेंट है. सो ये आरएनए कंटिन्यूसली बनता जा रहा है और ये आरएनए क्या करेगा? इट विल इंट्रैप द एक्स क्रोमोसोम. ठीक है, ऑब्वियसली इसने अब जौन सा कुछ ना कुछ तो करना है, ये बन रहा है तो वो साइज इसका जौन सा इंक्रीज कर रहा है, तो ये एक्स क्रोमोसोम इनकेज करेगा, इंट्रैप करेगा एक्स क्रोमोसोम को. एंड जब ये एक्स क्रोमोसोम को इंट्रैप करता है तो उसको ये कंडेंस कर देता है. ठीक है, उस पर प्रेशर एक्सर्ट करता है जिसकी वजह से एक्स क्रोमोसोम कंडेंस्ड फॉर्म में आता है, एंड दैट लीड्स टू एक्स क्रोमोसोम इनएक्टिवेशन. Other genes that are present in Zik region are the T6 and ZIT. T6 is present on antisense strand and ZIST is present on sense strand. The function of T6 and ZIST are opposite to each other. Whenever T6 express, X chromosome remain activated. So T6 के expression से X activated रहता है और ZIST के expression से X inactive हो गया. So when ZIST express, X chromosome is inactivated. Transcription of ZIST is at low level on all X chromosomes. Obviously दोनों X chromosome जो फीमेल में प्रेजेंट हैं उन पे ये सारे जीन्स भी प्रेजेंट हैं. तो दोनों X क्रोमोसोम पे जिस्ट की ट्रांसक्रिप्शन हो रही है लेकिन लो लेवल पे है. और जब एक दफा इनएक्टिवेशन का डिसीजन ले लिया गया at the blastocyst stage, then transcription continues and enhanced on inactivated X only. Transcription of ZIST basically inactivated X पे further on जौन सा है वो पास होती है, जबकि जो एक्टिवेटेड X है वहां पे T6 जीन अपने आप को एक्सप्रेस करता है और ZIST वहां पे जौन सा है वो इनएक्टिव हो जाएगा. So that's all about the inactivation process and the dosage compensation.